now that we understand how neurotransmitters activate the receptors and what the receptors actually do, we can talk about what goes on in the postsynaptic cell. So especially in the central nervous system, the effect of any given neurotransmitter on a postsynaptic cell can be very complicated. So the first question is, what kind of neurotransmitter is released? And then what kind of receptor do they bind to? So if it's a receptor that acts as a neurotransmitter gated cation channel, usually sodium channel, then it will create an EPSP. If it's an anion channel, it will usually create an IPSP. But that doesn't necessarily tell you what the postsynaptic cell will do, because really the question is, if we're talking about most neurons in the central nervous system most of the time, we want to know what will be the effect of that cell on the production of action potentials, because in order for a neuron to transmit the signal from its presynaptic cell, it needs to produce an action potential of its own that then goes down its axon and then propagates forward. So to understand what's going on in the postsynaptic cell, you have to realize that, first of all, these signals can come from different parts of the cell. So you can have synapses along the dendrite, along the cell body, at the axon itself. And you have to keep in mind that any given neuron can receive inputs or form synapses with, in some cases, hundreds of presynaptic cells. So at any given time, any one cell could be getting input from many different cells at once. Some of those are excitatory, some of those are inhibitory, and the size of the EPSP that's produced by any given synapse can vary by the number of receptors that are activated, the number of neurotransmitter molecules that are released, and a bunch of different factors. So this is some of these things we can quantify, and some of them sort of add up relatively simply. So, for example, here we're looking at a single neuron postsynaptic cell with a single presynaptic axon connecting with it, and we're recording the membrane potential. So here we're recording the membrane potential over time from this postsynaptic cell. And so if this is an excitatory synapse, and again, you can determine if a given synapse is excitatory this way. So if it's excitatory, then when the presynaptic neuron produces an action potential, then the postsynaptic neuron should experience this transient depolarization. Now, importantly, and this is something that people get confused a lot, the EPSP is not the same thing as the action potential. So an action potential, by definition, is only going to occur in the presynaptic cell traveling along the axon, and it's driven by the opening of voltage-gated sodium channels. The EPSP, by definition, occurs in the postsynaptic cell, and it's driven by inward current from the opening of neurotransmitter-gated ion channels. So when we see a change in membrane potential like this, again, it's not going to act like the action potential. It's not a spike. It's kind of gradual because it depends on these channels opening at sort of at random, not all at once, and they sort of gradually close as the neurotransmitter is removed. So there's no undershoot. There's not a real distinct falling phase. So that's the typical shape of an EPSP. Also, and then we'll talk about the position too. The location that you're measuring from also matters. But another thing that can affect the EPSP is if there are multiple synapses active at the same time. So over here, we're looking at the same neuron. We're recording, again, the membrane potential from the cell body. But this time, there are three synapses, or synapses from three different neurons, and all three are activated at the same time. So all three basically produce an action potential that travels down each axon and hits each synapse at about the same time. And what you see when that happens is that the size of the EPSP is greater. So again, if this was roughly the size of the EPSP of just the single synapse, this now is the increase in the postsynaptic potential caused by multiple synapses being activated simultaneously. This is called spatial summation. And it's simply the fact that now all three of these synapses, at all three of these synapses, you have a bunch of probably sodium channels opening, sodium currents coming in, and they all get to the cell body at about the same time. So they all cause a greater increase in the postsynaptic potential. Meanwhile, if you have, instead of three different synapses, just one synapse, but this time three different action potentials all arriving very close together, in other words, within just a few milliseconds of each other, then their EPSPs will also add on top of each other. So this would be the EPSP from the first action potential. This is the EPSP from the second, and it's larger because it started before the second one. The second one started before the first one really ended. So that means that the increased in membrane depolarization from the second one sort of stacked on top of the first one. And then the third one arrives before the second one is over. And so each successive postsynaptic potential kind of builds on the one before it. And that's called temporal summation. And so in both cases, you have the combination of multiple action potentials all having different effects on the postsynaptic cell. And again, all of these are going to have the potential to create an action potential in the postsynaptic cell. But remember that you can only have an action potential if the membrane is depolarized enough at this spot right here, which is, for most cells, this is the spike initiation zone, because this is where the voltage-gated sodium channels are going to be that will trigger an action potential in this cell. So the EPSP has to travel along this dendrite down through the cell body and get to the spike initiation zone and be large enough to be over threshold. Because remember, the threshold for triggering an action potential is about minus 40 millivolts. If the resting potential is about minus 65, then the peak, the magnitude of that EPSP needs to be roughly 25 millivolts, because that will bring the membrane potential up to negative 40 at the axon hillock or the spike initiation zone, if that happens to be the axon hillock, and you will get an action potential in that postsynaptic cell. So one way that one cell could increase the chances of generating an action potential in the postsynaptic cell would be to send, instead of just one action potential, a train of action potentials. Because let's say that right here is minus 45. What that means is that by itself, the single synapse would be unable to generate an action potential because it doesn't cross that threshold. But if you have two or three action potentials in a row and they stack on top of each other, that might be enough to trigger an action potential in the postsynaptic cell. Or you could have this situation where two or three synapses working together could trigger an action potential in the postsynaptic cell. And so this is actually really important, because this is actually how neurons can sort of act or process information or act as little computers. Because if you know how computers work, they have logic gates. So little circuits that can detect two different signals and, for example, only create a response if both inputs are on at the same time. That's called an AND gate. And the spatial summation, for example, is sort of a biological mechanism for an AND gate. Essentially, if you have two or more inputs active at the same time, they're more likely to trigger a response in the postsynaptic cell than if one of them was active alone. So this is the basic building blocks of a neural circuit. And another important factor that determines whether or not an EPSP triggers an action potential is where it falls along the dendritic tree, or where it falls within the cell body. So again, here we're looking at a cell where this is one of these big pyramidal neurons. It has this long apical dendrite, and so the synapse is way up here at this position on the apical dendrite, which means the EPSP has to travel And now, importantly, EPSPs don't travel the way action potentials travel. So that's another difference between action potentials and EPSPs. The action potential is kind of self-propagating. So as one voltage-gated sodium channel opens, it triggers the opening of its neighbors. But EPSPs don't work that way. An EPSP is basically just a passive current flow along the cytoplasm of the dendrite. There are no voltage-gated sodium channels, or not enough, to create an action potential that will keep the EPSP going. So EPSP just travels through the cytoplasm as passive current flow. But again, just like with an axon, passive current flow through the cytosol has the ability to leak out. So all along the dendrite and all throughout the cell body, there can be ion channels that will let current leave, and that will cause the EPSP to degrade. So let's say this is a dendritic cable, just somewhere along the dendritic arbor, doesn't matter where. And we've stuck an electrode in here, and we're injecting current, which means that we are depolarizing the membrane at this position. So if we measure the membrane potential at this position, we will see, let's say this is measuring it in terms of percentage of depolarization at the origin. So that's the peak. That's where the membrane depolarization is largest. And then if we measure at other positions along the cable, you will see that the magnitude of that depolarization gets smaller and smaller as you move away from that starting point in both directions. And the reason, again, is that current is just leaking out of that cable through ion channels. So it's like water going through a leaky garden hose. But again, there are no voltage-gated sodium channels to sort of restart or reinvigorate essentially the EPSP, and there's no myelin. So dendrites do not have myelin. There's nothing to stop that current from leaking out. So what it means is that if you measure an EPSP from a synapse, as it gets further and further away from that synapse, it becomes weaker and weaker. And we can, in fact, measure that. So it turns out that it degrades, the EPSP becomes smaller exponentially. So this formula here kind of describes the relationship between the membrane potential with distance. So the membrane Vx here means the membrane potential at any given position along the membrane x distance, and whatever units you want to use, from the starting point. So whatever starting point you pick. And so let's say if we're concerned with the distance from the postsynaptic membrane, then we can choose the postsynaptic membrane as our starting point, and we call that whatever the membrane potential is at that point, V0. So V at any point x is going to be proportional to V0, but it's going to degrade exponentially. So we multiply it by e to this factor negative x over this value lambda, and lambda is just a constant called the dendritic length constant, which is a property of the dendrite. So we call it a constant, but actually it's not constant. It can fluctuate with properties of the dendrite. So again, just like with axons, thicker dendrites have a larger length constant, whereas than thinner ones do. Dendrites that have a lot of ion channels that are open will have a shorter dendritic length constant because there's more current to leak out. So another way to think about the length constant is that if x is lambda, in other words, when you're measuring at a distance that's equal to lambda away from your starting point, then the membrane potential, you can just plug x or lambda into x up here. So V at point lambda is just V0 over E, or times e to the negative 1. And 1 over E is about 0.37. So another way to say it is that the membrane potential at the length constant is going to be about 37% of whatever it was when you started. And so that's just another way of describing the length constant. But like I said, the length constant is a function of the properties of the dendrite itself. And so in fact, the shape of the dendrite matters, the membrane composition of the dendrite matters, and of course the distance of the dendrite or the distance of the postsynaptic position from the cell matters too. Because again, if the EPSP becomes weaker as it gets further away from the synapse, then synapses that are farther away from the spike initiation zone are going to be less likely or have a harder time generating an action potential by themselves than those that are closer up. So closer synapses that are positioned closer to the spike initiation zone are going to be more effective at driving action potentials than those that are further away. And so that brings us to then inhibition. We can talk about inhibitory synapses the same way we talk about excitatory synapses. So inhibitory synapses work generally by letting current out of the cell. So excitatory synapses are generally characterized by cation channels. That means that channels that let positive current into the cell, whereas inhibitory synapses are either characterized by anion channels, so usually chloride channels that let chloride in, and that's the same thing as letting positive current out. So if you let a negative ion into the cell, that's the same thing as letting a positive charge leave. Or there are some inhibitory synapses or inhibitory receptors that are actually potassium channels. So potassium channels let potassium potassium out, and that will have the same effect. It will lower the membrane potential. So if you have an excitatory synapse like this, so you have an excitatory synapse at some distance along the dendrite, and it's active, it's going to create an EPSP. If you record right next to the synapse, you will see a relatively large EPSP. But if you record from that same cell closer to the cell body, and again, this axon hillock is where you need the action, where the action potential is going to originate, you might see that the EPSP is much weaker, but it's still there. Now, maybe this is enough by itself to generate an action potential, maybe it's not. But at least it exists. But if you have a position that is closer to the axon hillock than the excitatory synapse, so right here, for example, you have an inhibitory synapse. If that inhibitory synapse is active, so up here it's inactive, so this is the inactive synapse, so it's not doing anything. Down here it's active, so that means that there's an inhibitory neurotransmitter being released from this synapse. It's activating inhibitory receptors, which means probably chloride channels. Chloride's coming in, and that's lowering the membrane potential. And usually that has the effect, again, because typically for most cells, the equilibrium potential for chloride is pretty close to the resting potential. So you may not even see the membrane potential become negative. What you would instead see is at this position, so if this electrode will say that even though there's this big EPSP that's coming from upstream, at the downstream electrode, you see nothing. You don't see any change in membrane potential at all, because that influx of chloride has effectively completely canceled out, or any positive current caused by the influx of sodium is being canceled out by chloride coming in down here at this inhibitory synapse. And so from the cell body's point of view, there was no EPSP. It never got to the axon hillock, and so no action potential will be generated. So this is how inhibitory synapses work. But what's important is that the position of the synapse also matters. So if these were flip-flopped, if you had the excitatory synapse further away than if the excitatory synapse was closer than the inhibitory synapse, so if this synapse was, let's say, down here, for example, on the cell body or close to the axon hillock, then it would have much more of an effect. It would be much more likely to trigger an action potential, and in fact, the inhibitory synapse wouldn't really have much of an effect at all. So the location of the synapse on the postsynaptic cell is important. Now, one last thing to say about synapses before we finish this chapter is this idea of modulation. So up until now, we've been talking about excitatory and inhibitory synapses. So again, excitatory synapses by definition produce EPSPs. Those are temporary depolarizations of the membrane that have the ability to produce action potentials. Inhibitory synapses make IPSPs. Those are temporary hyperpolarizations of the membrane that prevent action potentials. And so neurotransmitters that activate ion channels are generally either one of those two, depending on the type of channel. Modulatory synapses or modulatory receptors, on the other hand, are more complicated because, again, they activate those metabotropic receptors, or the G protein coupled receptors. And it's not as simple to call them excitatory or inhibitory. Generally, they can change the membrane potential, but they don't necessarily directly affect the activity of the neuron. What they usually do is modulate the ability of other either excitatory or inhibitory synapses to do their job. And just as an example, again, there are lots of different examples of these, and we're not going to go through all of them, but this is just one example. So this is the pathway for one of the receptors for norepinephrine. So norepinephrine is one of the biogenic amine neurotransmitters, and it in fact doesn't even have any ion channel receptors associated with it. It only has metabotropic receptors. There are multiple receptors for norepinephrine. This particular one we're talking about is called the beta receptor, or strictly it should be the beta norepinephrine receptor, but usually it's just abbreviated the beta receptor. So when norepinephrine binds to the beta receptor, it activates a G protein. So remember, the G protein is a separate structure from the receptor. So this is the receptor, this is the protein. And G proteins, we'll talk more about them later, but suffice to say for now that when the receptor activates the G protein, this particular G protein in most cells activates an enzyme called adenylyl cyclase. So adenylyl cyclase is activated by G proteins, by a specific G protein, in this case that is in turn activated by the beta receptor. And adenylyl cyclase, again, it's an enzyme, and the reaction it catalyzes is the conversion of a molecule of ATP into a molecule of cyclic AMP. So ATP, we already know what that is. It's adenosine triphosphate. It's found in all cells, and mostly we think of it as being used for energy, and that is what it does most of the time. But adenylyl cyclase, instead of, actually it does use the energy from ATP, but in the process of using it, instead of making ADP, which is what most enzymes that use ATP do, it converts it into a different molecule called cyclic AMP, or cyclic adenosine monophosphate. So it actually takes off two phosphates and then connects one end of the phosphate of the AMP back to itself to make a ring, which is why it's called cyclic AMP. And cyclic AMP is an example of a second messenger. So this enzyme will make a whole bunch of cyclic AMP. As long as ATP is present and as long as that G protein is active, adenylyl cyclase will just crank out cyclic AMP all day. And again, we call it the second messenger because this is the signal that's going to propagate this cascade within the cell. The norepinephrine essentially was the first messenger. That's just where the term comes from. And so cyclic AMP then does a whole bunch of things. It actually activates multiple pathways, but one of the things it does is it activates a different enzyme called protein kinase A. Now, a protein kinase is a special type of enzyme that works by adding phosphate groups to other proteins. So a phosphate is just a phosphorus atom with 4 oxygens attached, and kinases are enzymes that take those phosphates and add them to other proteins. Now they're specific, so protein kinases will only phosphorylate certain other proteins, and then the effect of phosphorylation depends on the protein. So some proteins become more activated when they're phosphorylated, some become less active. One of the proteins that protein kinase A phosphorylates is a special kind of potassium channel. So potassium channels are exactly what you think they are. They're just those leak channels that most of the time are open, and they let potassium out. So if there's potassium present and there's an open potassium channel, potassium will leave, and that will keep the membrane potential low or hyperpolarized, because potassium leaving makes the cell more negative. But it turns out that when phosphorylated, the potassium channels close. So the effect of phosphorylation is to reduce the potassium current leaving the cell, which basically means the membrane potential will not be as negative. Again, this is not the same thing as an EPSP because you don't see a sort of transient depolarization. Instead, it effectively sort of raises the resting potential somewhat, which in turn means that any subsequent excitatory synapse, any other EPSP that comes down this cell, will be more likely to trigger an action potential. So that's what modulation means. You're not directly increasing or decreasing the activity of the cell necessarily. You're just making it more likely that some other synapse will be able to activate the cell. So again, it just sort of modulates the baseline activity of the cell. And that does it for this chapter. The next chapter, we will talk about neurotransmitter systems.